So taking everybody's request into consideration, there should be a little picture of me on here so you can watch my face and see my lips move while I'm narrating this. And I am going to do my best to put the writing back on here. Bear with me because I'm sure that it's going to take me a little while to get control of that. Okay, ready to go. Now we're going to talk about regulating enzymes. So enzymes need to be regulated because you don't want them going on all the time. There's homeostasis in the body, which is just a fancy term for maintaining stable conditions, which means things have to work. Things have to turn off when they're supposed to turn off. Things are supposed to work when they're supposed to work. You want enzymes working maybe in your small intestines when food comes in. You want to synthesize amino acids if you haven't been eating something and you really need that amino acid to make a protein. Your enzymes have to do work for all different kinds of reasons. So this section is going to talk about how our cells regulate enzymes, maybe how some things could interfere with them, and all that stuff. So, moving down. Okay. We can have inhibitors that can bind and slow reactions down or completely turn them off. So I'm just going to jump ahead. The first one that we'll talk about is an irreversible inhibition. So this one's pretty awful. This would be almost like a poison. This is if something binds, it's going to literally kill the enzyme and will never work again. There are some, got to clear those off. There are some chemicals, like some insecticides that work like this. This is an example of a chemical, and this is the inhibitor right here called oops, didn't want to do that. DIPF. And this little molecule, so it's just an organic ring, can bind in the active site. And if it binds in the active site of the enzyme, then our normal ligand that the enzyme would work on is not allowed to bind. And this one actually binds there, and it's irreversible. It totally decommissions that enzyme. It's useless. So you're going to find a lot of toxins and poisons that do it like this. Oops. Okay. More commonly found would be competitive inhibitors and non-competitive inhibitors. And these guys, the competitive inhibitor is something small that has the ability to compete with that natural substrate for the active site. So if my enzyme is shaped like this with the active site in there, a competitive inhibitor, and there's going to be a picture coming up too, would kind of go over here and block your substrate from being able to get in there. So it competes with it. These have a natural on-off rate. So any chemical binding, there's an affinity constant. You might have talked about that in chemistry. So it kind of comes on and comes off. If it has a high affinity, it kind of stays on, stays on, stays on, and only falls off once in a while. So some of these inhibitors have different levels of affinity for it. And sometimes your substrate could compete it out and get in there. On the other hand, there are non-competitive inhibitors, and these guys bind somewhere else on the enzyme, so maybe out over here, and it changes the shape of your enzyme so that your substrate can't get in there. So let's actually go look at a real picture of this. So here's our competitive inhibitor, and this one is going to block the active site so your substrate can't get in. Pretty obvious. Something's there, your substrate can't get in, your enzyme can't work. A non-competitive inhibitor, on the other hand, is one that binds somewhere else. So this guy is going to bind on this other side of the protein. Remember, proteins have 3D shape. And it changes the active site. It kind of closes it up. They show it almost like a little flower, kind of closing up over here. When this non-competitive inhibitor, and this could be any kind of a chemical structure, binds, it shuts it down, and now my substrate can't find in. These guys sometimes are harder to get rid of because that stays stuck, and there's no amount of substrate you could put in that's ever going to make this work. In the competitive inhibitor situation, you could flood your system with substrate and then finally maybe knock this guy out. Again, it's still pretty tough to do. So those are two ways that you can regulate an enzyme. Another way that's more of the way you might control an enzyme in a pathway in a cell is called allosteric regulation. And this one is kind of like the non-competitive inhibitor. People get them confused sometimes, but basically it's a non-substrate molecule, so it's some other protein or probably a little chemical actually, and it binds somewhere other than the active site. But in this case, maybe it goes on and off, and it's going to change whether your substrate can bind. Let's just move to a picture. I think it'll help. Oh, I'll leave that there. So let's start with our inactive enzyme up here. It has a site that can get regulated. If that regulator binds, it opens up the active site. See, over here it was closed, and over here now it's open, and it can accept the substrate, and we have something happen. There's another way that this might happen. So here's our inactive enzyme, and sometimes a phosphate gets added to a protein. Remember phosphates, the POOOs with the negative charges? And if that phosphate's there, it also opens it up. We're going to see this happen when we start talking about a topic called, see, I can't write, signal transduction. I'm not even going to try. Signal transduction. This is something that we're going to talk about, how the cell receives a piece of information, a signal on the outside, and then through a cascade of enzyme steps can make something happen basically on the inside of the cell. We'll be talking about that in a couple weeks. So protein kinases are special kinds of enzymes that add phosphates to things, the phosphorylating allosteric sites, and that's essentially what we're talking about here. So a protein kinase is this other thing that's involved, adds a phosphate to this, activates it. There's a flip side that you could remove a phosphate and then deactivate it, so it sort of can flip back and forth. So why would you want to do that? Well, maybe sometimes you don't want that enzyme working. Maybe you don't want to be telling a cell a piece of information, like telling it to grow or telling it to divide. So you want to control that, and you only want to turn it on sometimes. Similarly, there's times when enzymes make products, and I'm going to just jump to the picture because I think it'll help. So here's the amino acid threonine, a simple molecule. It turns out that if you don't have certain food in your diet, you can make some amino acids. This is an amino acid called isoleucine. So you can take the amino acid threonine, and through, it shows you here, 5 steps, you can turn it into isoleucine. So what if you've got tons of isoleucine hanging around? Oops, didn't want to do that. What you want is you want to shut this pathway off. So isoleucine comes back and negatively inhibits. This is feedback inhibition. It shuts the pathway down. Why would I bother to do all these steps if I have a ton of isoleucine around and I can shut this all off? So there are times that you might want an enzyme. That's what this green arrow would be representing, is that an enzyme is making this reaction take place. And sometimes you're going to want to turn that reaction off. So isoleucine will come back, maybe bind allosterically, and turn it off. And then we did a little bit of this experiment in class the other day. We know that pH can affect enzyme activities. So we took amylase from our spit and we added HCl to it. And we basically turned that enzyme off. And we turned it off because those hydrogen ions interfere with the structure of the enzyme. Similarly, you can interfere with the structure of an enzyme using a base. So we know that certain enzymes have very specific pHs where they work. So we know that we digest food in the food gets mushed up and put in our stomach. There's a lot of acid there. There's HCl there. And then it enters the small intestine. But there are enzymes that work in our stomach. Pepsin is an enzyme that breaks up proteins in our stomach. So if you have hamburger, you're going to have pepsin working. And it actually has an optimal pH somewhere down near acid pH that's in the stomach, a little between 1 and 2. But then we already did the amylase experiment, and we know that that enzyme works in our saliva, and it has an optimal pH. It's not on this thing, but amylase has an optimal pH of 7. So they're also showing you that there are other enzymes that have an optimal pH that's different, and they would die if they were in the stomach. They would be too acidic. Similarly, temperature can affect an enzyme's function. So at some point, increasing a temperature actually can increase the rate, and this is purely kinetics. If you increase the temperature, molecules are bumping into each other a lot more. You're going to have an enzyme finding its substrate and bumping into it more frequently, and therefore you're going to have your substrate, the reaction rate increasing. But you hit a point with the temperature that you'll break the hydrogen bonds in the enzyme, and then your active site falls apart, it basically unravels, your alpha helices unravel, your beta pleated sheets unravel, and the structure of a protein kind of falls apart. Maybe just a little bit even, so that the active site's not quite as perfect as it used to be, and then your enzyme won't work anymore down here. And we'll investigate this in the enzyme lab that we do when we come back from outdoor education. And actually, that's where I wanted to stop.